Hello and welcome back to our second video on photosynthesis from Mr. Carter Science Specials. This video will cover two areas. It will cover the starch test and it will cover the rates of photosynthesis reactions, how you describe and how you explain those. As always, please look out for the curriculum references or syllabus references so you can follow along where you are in your syllabus. So first thing, how do we know that a plant has been photosynthesizing? Well, before I carry on with this, just pause the video now, see if you can work out the two missing gaps on this slide, and then I'll go through the answers in a moment. So how do we know if a plant's been photosynthesizing? Well, during photosynthesis, plants produce glucose. But plants cannot store glucose because it's soluble. It has a big osmotic effect on the cell. So what that means is if a plant begins to accumulate glucose, then that gives it a very high concentration. And due to osmosis, water will flow in from a low concentration to the high concentration. And this puts a large pressure on the cell. It will cause plasmolysis, and possibly even, despite having the cell wall, the plant cells could be at risk of bursting as well. So plants cannot store glucose because it's soluble and it has an osmotic effect. So instead, the plant cells join the glucose molecules together and they form a long chain molecule called starch. And starch is a long chain of glucose molecules all joined together. And we can see that in this diagram here. Take the four glucose molecules, join them together, and you produce one molecule of starch. And starch is insoluble. So starch is very good for storage because it's insoluble and it does not have any osmotic effect on the cell. So if a plant is photosynthesizing, it will be making the glucose, but shortly afterwards it will also be making starch. So if we test a leaf and it has starch, it must be photosynthesizing. Now this is really important because the test for starch is a relatively easy test to perform on a leaf, but the test for glucose is much harder and far less reliable on a plant cell. So how do we carry out this starch test? Well, again, pause the video, see if you can look at the 4 steps here, see if you can use the words in the box so that you can explain and describe how to carry out the starch test yourself. So I'll show you the answers in a moment, but this is really important for your exams because you can easily get 4 or 5 marks on a question which says describe how to carry out the starch test. Often they will add in a bit of safety and say describe how to safely carry out the starch test. So you may not pick up marks for just the starch test, but you'll pick up the marks where you've mentioned safety measures. So while you're carrying out this, you should always have goggles on because you're going to be using various chemicals that can affect your eyes, and you're also going to be using Bunsen burners. And when we come to it, I'll talk about the other safety measures as we get to them. So the first thing to do is to place the leaf in very hot boiling water. Now you may have boiled your water by using a Bunsen burner and heating from a tripod. In my classroom, I tend to just use a kettle because it's a bit faster and it's safer because I don't have the Bunsen burners out. So place the leaf in very hot boiling water for 30 seconds and this will soften the leaf and kill the cytoplasm. The next step is we need to remove the chlorophyll from the leaf because chlorophyll interferes with our ability to see the changing colour of the iodine. So we're going to place the leaf in a test tube with some warm alcohol for about 5 minutes, and we're going to keep that alcohol warm by warming it in a hot water bath. You will often find that you'll use exactly the same beaker with the hot water because it's warm enough to boil the alcohol. Hot water, perhaps at say 90 degrees, will easily be higher than the temperature that alcohol needs to boil of around 78. But here's a safety tip. We must, if you've got Bunsen burners, turn off all flames because alcohol is flammable. Again, that's an easy mark to pick up in an exam. So this will actually remove the chlorophyll and the alcohol will turn green. So after 5 minutes or so, you'll find that you've got a large amount of green alcohol in your test tube. The longer you can actually leave it in, the better because the more chlorophyll is removed and the clearer you will see the colour change at the end. So the alcohol removes the chlorophyll and the alcohol turns green, but the leaf therefore has lost its chlorophyll and turns white. But that leaf will also be very, very brittle. So we need to soften that leaf again, and we do that just by taking the leaf and washing it in the hot water in the beaker for around 10 seconds just to soften the leaf again. We then need to place the leaf on a tile and add a few drops of yellow iodine. And that's how you carry out a starch test. You need to know and memorise all 4 steps. But you also need to know the results. What are we going to look at? So if starch is present, our yellow iodine will turn blue-black. So if a leaf that looks like this does not have starch because the iodine is still noticeably a yellow colour, but a leaf that looks like this has a distinctive characteristic blue-black colour, this is a leaf that has starch. So we can use this test now to find out about a range of conditions that will be needed for photosynthesis to occur. So the first thing you may well have tested in your classroom is whether light is needed for photosynthesis to occur. And the classic experiment for that is to use a leaf with a strip of black paper covering one section of that leaf. And you will generally get this kind of result, where the area where we have no light at all is yellow because it has had no photosynthesis occurring. So you might be asked to label this in an exam. So the first thing you need to say is that the iodine is yellow, and that means there is no starch and therefore no photosynthesis. The opposite is true, of course, in these 2 areas here, and in these 2 areas the iodine is now blue-black, and that means that we have starch, and so we have had photosynthesis. So what's the conclusion? Well, the conclusion is very simple. Areas with no light have no starch. Areas with light have starch. Therefore, light is needed for photosynthesis. And you may have done a similar experiment with a type of plant called a variegated leaf. And we've got an example of a variegated leaf at the start of this experiment here. And variegated means it has different colours. And those colours are caused by having different amounts of chlorophyll at different areas of the leaf. So in this leaf, we've got a large amount of chlorophyll in the centre of the leaf, but no chlorophyll around the edge of the leaf. So this is a variegated leaf. So leave it out in the light for a number of hours. Take it back inside. Again, what do the results show us? Well, here's a set of results, and you may well have to label this in an exam, and it will be very similar labels to the labels we've already written previously with light. The conclusion is that areas with no chlorophyll have no starch, and areas with chlorophyll have starch. Therefore, chlorophyll is needed for photosynthesis. The final experiment you may have carried out is to do with carbon dioxide, and in this case we would need 2 separate plants, both covered in a set of transparent plastic, but they have slightly different chemicals inside them. For example, this particular plant here, plant B, has soda lime, and soda lime absorbs carbon dioxide from the air. So in this particular plant, the leaves will have no access to any carbon dioxide because it's all been absorbed into the soda lime. In plant A, we have a large amount of sodium hydrogen carbonate, and that's a chemical that will give out carbon dioxide. So the leaves have got ready access to carbon dioxide. At the bottom, we can see the results of this. We have in plant A a blue-black leaf, which clearly means that we have some starch made in plant A, so we must have been carrying out photosynthesis. And in plant B, there is no starch present because the iodine has stayed yellow. So there's been no photosynthesis. So what's the conclusion? Well, leaves with no carbon dioxide have no starch, and leaves with carbon dioxide have starch. Therefore, carbon dioxide is needed for photosynthesis. And we can use this information to begin to build up our word equation for photosynthesis. We've just proved that we must have carbon dioxide in that word equation. We know that we've got to join that with some water, and we make the glucose that we test for, and we make the oxygen. But we can't have this equation. It will not happen unless we have a source of energy, which we proved before was light, and a particular molecule for this to absorb the energy, which is called chlorophyll. So we've begun to prove the photosynthesis equation using these simple experiments. The other thing to consider with these experiments is that all the plants used will have all started out being destarched. They will have all had no starch in their leaves. The way that you achieve this is that you take these plants and you place them in the dark for around about 48 hours or so, and that means that all the starch reserves will get used up and broken down, and the plant won't be able to photosynthesize, so it won't be able to produce any more starch. So it's really important we start with destarched leaves so that we prove that any starch that we find is due to photosynthesis and not due to some other problem. So, what factors might affect the rates of photosynthesis? Well, we can use this equipment to investigate any factor that we think might affect the rate of photosynthesis, and then we can prove that one, it does affect the rate of photosynthesis, and then we can begin to describe how and explain how that particular factor affects the rate of photosynthesis. So how can we do that? Well, we need to take this kind of equipment. First of all, we need plant that can photosynthesize. Typically a pondweed is used. We place that into a conical flask full of some water, and within that we probably put some sodium carbonate as a source of carbon dioxide for the plant to be using, and we make certain that we have a lamp as a source of light. And we sit and we watch and we wait. We should begin to see after a few moments little bubbles of oxygen gas produced. Now at that point you can either begin to count how many bubbles are produced per minute, or you might have a system set up to a syringe which can measure the volume of oxygen produced per minute. And it's that per minute that's really important because we're looking at rates. So we're always looking at the amount of oxygen produced, and that would typically be either the number of bubbles or the volume per minute, and that will tell us our rate. Now, because you're not going to produce a large amount of oxygen, you might have to leave this equipment running for maybe 10 minutes. So whatever volume you manage to get, volume of oxygen, you will have to divide that by 10 to tell you the volume of oxygen per minute, because we left it for 10 minutes. So there's 3 different factors that can affect the rate of photosynthesis, and those factors are light intensity, carbon dioxide and temperature, and they will all affect the rate or the speed at which the plant cells can make glucose and starch, and the amount of oxygen that's produced. We always measure the oxygen because it's much easier to measure volumes of oxygen or masses of oxygen than it is glucose and starch. When you do these experiments where one by one you change the independent variable, for example the independent variable being light, you will then produce this graph, or if you have the independent variable as carbon dioxide, you'd have this graph, or the independent variable as temperature, you'd have the third graph. You must be able to describe and explain these 3 graphs ready for your exams, and you need to know the difference between the two key words there of describing and explaining. So we're going to go through each graph one by one and make certain that you can describe and explain each graph. So the first factor we'll look at is light intensity, and here we've got a gap fill exercise to see whether you can describe and then explain the graphs in front of you. So pause the video for a moment or two, write these down, sketch out the graph and see whether you can describe and explain these before I go through the answers. So here are the answers. Light intensity. You'll notice that each graph has two distinct areas, both of which need to be described and explained in different ways. So for each of the three graphs we're going through, I've got two different paragraphs to talk about two different areas of a graph. So the first paragraph will always mention for the first part of the graph, and the second paragraph will be to describe and explain the second part of the graph. So let's look at this graph when we've changed the light intensity. First of all, we check at the bottom that we have the correct labelled axes, that the independent variable has gone on the bottom, and that our dependent variable, the thing we're measuring, the rate of photosynthesis, has gone on the side. So as the light intensity increases, the rate of photosynthesis increases. And we can see that clearly in this first section. As light intensity increases, the rate of photosynthesis increases. And this is because light intensity is what's called a limiting factor. And that means an increase in light intensity will allow a plant to make more glucose and oxygen. Well, this is a fairly obvious claim. More light, more energy, therefore a greater rate of reaction. However, in the second part of our graph here, that's no longer true, because as the light intensity increases, the rate of photosynthesis stays the same. We do not get an increase anymore. And all I've done there is describe. As the light intensity increases, the rate of photosynthesis stays the same. That's describing, but we might have to explain why. And explaining is always where we're trying to explain and say why something changes. So as the light intensity continues to increase, the rate of photosynthesis stays the same. And this is because light intensity is no longer the limiting factor. Something else is slowing down the rate of reaction. In this case, it could be the amount of carbon dioxide or the temperature that we do not have enough of. For example, if it's the amount of carbon dioxide that's limiting things here, then if we don't have enough carbon dioxide, we can't create new glucose molecules. We have to have a constant high supply of carbon dioxide to make certain we have more and more glucose molecules. If it is temperature, that's the limiting factor. Perhaps we've got plenty of light, plenty of carbon dioxide, but it's a cold day. We're looking at temperatures of maybe only 10 degrees, in which case those rates of reaction will not be going at a particularly fast speed. All the molecules involved will be moving quite slowly. So temperature will be limiting the rate of reaction at that point. So you need to remember, limiting factor is always the thing that slows down the chemical reaction. If we increase the limiting factor, we're going to increase the rate of reaction. So in the first section, light is the limiting factor, because if we increase it, we increase the rate of reaction. But in the second section here, light is no longer the limiting factor. Something else is: carbon dioxide concentration or temperature. So the next factor we're going to look at is carbon dioxide concentrations. Again, we've got a gap fill here, so just pause the video for a moment, have a think, write down the two paragraphs, sketch out the graph and see whether you can describe the effect of carbon dioxide on the rate of photosynthesis. So here are the answers. As the concentration of carbon dioxide increases, the rate of photosynthesis increases. There's the carbon dioxide increasing, there's the rate of photosynthesis increasing, and in this first section of the graph we get the classic positive correlation between the two. And this is because in this particular experiment, carbon dioxide is the limiting factor. The more carbon dioxide we have, this will allow a plant to make more glucose and starch and more oxygen. So if we gradually increase the amount of carbon dioxide that we give a plant, we're going to get more oxygen produced, more carbon and more glucose produced, and a faster rate of photosynthesis. But after a while, that's no longer true, because as the rate of carbon dioxide, as the concentration of carbon dioxide increases, the rate of photosynthesis stays the same. We've reached a plateau. So how do we describe and explain this? Well, we say, as the concentration of carbon dioxide continues to increase, the rate of photosynthesis stays the same. Carbon dioxide is no longer the limiting factor. Instead, the limiting factor will be the amount of light or the temperature that we've got the reaction going through. Light and temperature are now the limiting factors. The final factor that we're going to look at today is temperature. Again, we have a gap fill, so please pause the video, see if you can describe and explain what is happening in both areas of this graph, and I'll give you the answers in a moment. So as the temperature increases, the rate of photosynthesis will increase. And we can see that here and here. In this first section of the graph, as the temperature increases, the rate of photosynthesis increases up to an optimum temperature. Now that optimum temperature is typically around 40 degrees C for most plants, particularly plants that live in temperate zones of the Earth. And this is because temperature is our limiting factor. An increase in temperature allows the plant to make more glucose and more oxygen. And this is because of basic chemistry and basic kinetic theory. The higher the temperature, the faster the molecules are moving, the greater the likelihood of having a successful collision between all the molecules involved in photosynthesis. So the higher the temperature, the more successful collisions you're going to have and the more products you're going to make. However, after about 40 degrees C, this is no longer true. And that's because after 40 degrees C, as the temperature continues to increase, the rate of photosynthesis actually begins to decrease. And this is because all the enzymes that are present on a chlorophyll molecule that control photosynthesis have become denatured. So the carbon dioxide and the water can no longer be combined to make the glucose and oxygen. Therefore, the rate of reaction decreases massively. I hope you've enjoyed this video on photosynthesis, rates of photosynthesis and the starch test. Please like and share so that you will get notifications when the next video is released from the Mr. Carter Science Specials. Thank you very much for watching.